Hello, and welcome to another dive into our microbiology text by Tortora. We've come to chapter 17, the adaptive immune system. And last chapter was the innate immune system, in which all the responses are the same every time. And now we come to a system that remarkably produces a different response every time, depending on what particular antigen or foreign substances are coming into the body. There's our kickoff, beautiful micrograph of a phagocyte. Actually, in this case, it's a cancer cell about to get killed by a lymphocyte, a natural killer cell or something like that. Okay, so we're going to look at this specific immune response system. Right, we can put this in the context of some famous, historically important scientists. Pasteur observed immunity in chickens injected with weak pathogens, so that if they'd had a bout of some mild illness, then they would be immune later to other similar and related illnesses. Von Behring received the Nobel Prize for development of an antitoxin, which turns out has antibodies, which are defensive proteins produced by the adaptive immune system. Excuse me. And can bind to the toxins and prevent them from then binding to anything else. And then Ehrlich led to the identification of antibodies in serum. We'll say something about that descriptor serum at the very end of the chapter. So here is a crazy summary slide, which I think will be good for us. Let's just look through this a little bit together, and then hopefully some parts of it will stick in your mind as we're going through and looking at little sections of it one at a time as we go through the chapter. So here's what's kind of going to be happening. We have two arms or branches of adaptive immunity. Humoral immunity, which deals with extracellular antigens or foreign substances. And humoral means in the body fluids, and it's all about antibodies. Antibodies are defensive proteins that specifically recognize and stick to foreign glycoproteins, proteins, polysaccharides, anything foreign that comes into the body. Cellular immunity deals with intracellular antigens or foreign substances or infective agents, and it's all about cells that can go around looking for infected cells and killing those cells. So let's quickly just look through this roadmap. Antigens are any substance that can trigger or elicit an immune response. Something foreign comes in the body. If it starts up an adaptive immune response, it's an antigen. So in come some antigens, maybe some bacteria or viruses, and they have foreign proteins and polysaccharides and so forth on their surface. They come into the body and they bounce around among a whole bunch of B lymphocytes in lymphoid tissue, and eventually they'll bump into one whose unique B cell receptor recognizes or sticks to that particular chemical substance. Once that happens, the B cell gets activated, and it will multiply into a large, what's called a clone, a population of identical cells, just like when you had a clone of bacteria. A clone of B cells, some of which will mature into plasma cells, effector cells they're sometimes called, which can produce antibodies, and antibodies can go in the blood, the circulation, and body fluids, and stick to the antigens, and the phagocytes, once that happens, will gobble them up. Over here, we see a particular cell, a phagocytic cell perhaps, that has gobbled up or been infected with something, either consumed, gobbled up, or phagocytosed a bacterium, or was infected with a virus, or gobbled up a virus. And what it does is it displays fragments of those foreign substances on its surface, and a different type of lymphocyte, a T lymphocyte, will recognize the foreign antigen if it's the right T cell with the right receptor for that day's antigen, and then it will become activated, and let's follow down straight down. And once it's activated, it will mature into a cytotoxic T cell, which can kill other infected cells. So this cell down here at the bottom is not the same cell. It's an infected cell in your body, anywhere in your body. That cytotoxic T cell is going to circulate in your body and surveil, looking for any infected cells, and when it finds one, it'll kill it. So we have B cells that recognize an antigen of a particular type, and then they produce a whole bunch of clone cells that can produce a whole bunch of the identical antibody, which we know will recognize the antigen that's impacting our body today. Same thing with T cells. This antigen presenting cell, it's called, will circulate all around, bumping up against T cells after T cell after T cell until it finds the right one whose receptor can recognize the antigen that's here today, and it will become activated, and it has a specialized receptor. It will form a big clone, also a whole bunch of identical ones that will go off like an army into the body looking for infected cells with that same exact antigen that we already know is causing the trouble. Just to make sure that we don't make any mistakes. Oh, something weird happened here. My little blue box got kind of shifted out of position. Just so we don't make any mistakes, there's a separate type of T cell called a helper T cell. And that helper T cell is going to be activated in a similar way by an antigen presented on the surface of an antigen presenting cell. And that helper T cell has a unique T cell receptor as well. So again, the antigen presenting cell might bounce off a whole bunch of helper T cells till it finds one whose receptor recognizes that antigen. And when that happens, that T cell will become activated and it will produce some cytokines. Here's the important thing. Without those cytokines from a helper T cell, the B lymphocyte would never have gone on to proliferate and form a clone and plasma cells and memory cells, as we'll see later. It has to have that second signal from a helper T cell to say, yes, this is actually an infection. This is the real deal. This is not a mistake. We need to go ahead and make plasma cells and antibodies. Same thing with cytotoxic T cells that kill infected cells. Without some cytokines coming from that activated helper T cell, that cytotoxic T cell would never have proliferated into a clone of cells, an army of cells to go out on the road and kill infected cells. It would have stopped in its tracks and it would have never moved again. That's called anergy. So we have that safety net built in, that safety system where a second signal is required, co-stimulation, to get either one of humoral immunity or cellular immunity up and running. So again, humoral immunity, B cells and antibodies, cellular immunity, cytotoxic T cells, and killing infected cells who have antigens hidden down inside the cell. All right, so we can go on now and break this down into different parts and kind of hit it little by little and try to make some sense out of each one of those steps. So where do they come from? Lymphocytes, like all blood cells, come from the red bone marrow. And so the stem cells in the red bone marrow divide and produce progenitor cells, which will eventually result in the production of large numbers of B lymphocytes in the red bone marrow. And those B lymphocytes, once they are matured, we'll talk more about that later, they will go off and situate themselves in some lymphoid tissue, either the spleen or tonsils or lymph nodes, and that's where they live. They go to those organs and they stay there and wait for their antigen to come to them, which they can recognize. T lymphocytes are also produced in the red bone marrow, so this picture's a little odd. The arrow should have been coming from over here, from the red bone marrow, and then those cells do not go out to get to work yet. They're not ready. T cells go from the red bone marrow to the thymus gland, a gland under your rib cage, right over kind of over top of your heart and your esophagus and so forth. And that's where the cells mature and become prepared to go out, and they circulate through the body. T cells circulate around. They don't hang out and just stay someplace like B cells, because they're the kind of cells that need to bump into some event somewhere in your body and find out what's going on and potentially kill some infected cells. And those infected cells aren't going anywhere, so they need to go to where they are. So starting with humoral immunity, B lymphocytes are the ones that make antibodies, and they have all to do with humoral immunity. Cellular immunity is all about T cells and killing infected cells, intracellular antigens, hidden antigens, and again, T cells mature in the thymus. So what's an antigen once again? An antigen is a foreign substance. I should have put the word foreign in there. A foreign substance that elicits an adaptive immune response. It could be any chemical, really. But typically, it's a protein or glycoprotein or polysaccharide from a bacterium or a virus or a helminth or something like that, or a protozoan. Comes into the body. It looks bad. It looks foreign. and the body's going to know about it, right? We already know that. We know that macrophages and dendritic cells have toll-like receptors that recognize those things as foreign, gobble them up, process them, meaning digest them, and display the antigen on their surface, along with their MHCs. We'll talk about those in a second. Just an aside real quick, some chemicals in nature, which are foreign to your body, may be exposed, your body may be exposed to them, and on their own, they're not antigenic. They don't trigger an immune response. But occasionally, one will combine with a protein on one of your own cells or your own tissues, and that combination of that foreign chemical may be too small to start an immune response, but together with that protein, now it looks foreign, and you can get quite an unfortunate allergy thing going on. Like, for example, someone who gets poison ivy, that poison ivy oil from the plants combines with some protein in your skin, and T cells recognize that as being foreign and start getting all activated, releasing cytokines. Macrophages come in there, and all kinds of tissue damage happens, all because of that hapten that got in there and conjured up that kind of quasi-inappropriate immune response. All right, so here's an antibody. It's shaped like physically like a little letter Y. It has two like little prongs that are unique. Every antibody, every B cell makes a different antibody with a different type of tip on it that would recognize a different antigen. So here we have in this cartoon drawing one antibody that happened to be produced by a particular B cell that recognizes this triangle antigen. Here's another antibody that would have been produced by a different B cell, and it recognizes this little round humped antigen. So just the point is that every B cell has a unique antibody receptor that it uses to scope out antigens. And if it finds its antigen, then it makes a whole bunch of identical antibodies to that receptor and secretes them so that they can go off and take care of whatever bacteria are trying to colonize our body on that occasion. So that's just a picture of a hapten molecule coming into your skin like the poison ivy oil, combining with a carrier, so-called carrier, conjugate carrier, and causing trouble because it acts like an antigen then. The thing is, that's actually your own body's protein. Antibodies are called immunoglobulins. That's the nickname they have from way back when they were first discovering the plasma proteins and separating them from each other. They just called all the plasma proteins, they called them globulins, and they give different names to them: alpha globulins, beta globulins, gamma globulins happen to be the ones that they came up with for antibodies. So immunoglobulins or gamma globulins are what they're called. Valence just means how many little prongs that could recognize antigen does the antibody have, and that's called valence. The antibodies we saw in that picture have two. They have a valence of two. They have two separate binding sites. Here's how an antibody looks a little more up close. It has 4 peptide chains, 2 heavy chains, meaning it has a long stem, as well as this V part, and then 2 light chains which just span the V part, and they're disulfide linked. Sulfhydryl groups from cysteine residues can covalently attach to each other. Now we have a single multimer made out of 4 peptide chains. The tips, these variable regions they're called, the V stands for a variable region, the tips are unique. Every single antibody is a unique tip that's different, and it recognizes a different electrochemical property of some specific chemical, a specific sequence of amino acids in a peptide, and it will stick there and do its thing. They're produced during development by shuffling of a bunch of genes that make up the little segments of this variable tip, and so we get this random assemblage of amino acid sequences that are every single one of which is unique. So what is an epitope? Every antigen could be a large protein. It could have hundreds of amino acids, say, for example, a bacterial membrane protein or cell wall protein. It might only take a string of a handful of amino acids to form enough of a recognizable entity for an antibody to stick to it, or a B cell receptor or a T cell receptor to stick to it. That little bit of the protein that can, or polysaccharide or glycoprotein that can be recognized by an antibody is called an epitope or an antigenic determinant. In other words, a big protein could have quite a few places that different antibodies could recognize. And so whenever we talk about the things happening in this chapter, just remember it could be happening many times over in parallel, but we're just going to boil it down and make it sound like one single event for simplicity. And there's some actual electron force, atomic force microscopy, meant to say, that actually is showing us some antibodies, the little Ys. You can see them right there in real life. Actual molecules, proteins sitting there on a flat surface. Cool. So antibodies come in slightly different styles or flavors. This is an IgG, an immunoglobulin type G or class G, and this is the most prevalent kind of antibodies in the blood. 80% of, again, here it says 80% of serum antibodies. I think what they mean is plasma antibodies. In other words, in your blood isn't serum, it's plasma. And so there's 80% of the plasma antibodies. I guess they could mean, though, if you took out some of the blood and let it coagulate and centrifuged it down, then you'd have serum. Then if you checked, you'd have 80% IgG still, I guess. Okay. Anyway, these antibodies can activate complement or fix complement. They can circulate in the blood, in the lymph fluid, in the intestines. They cross the placenta. When a mother gets exposed to an antigen and has an adaptive immune response and makes antibodies, those antibodies then also protect the newborn because all those antibodies cross right over into the fetal circulation in utero. And then when the baby's born, it's got all the mother's antibodies. So it's pretty robust immunologically at that time. Antibodies enhance phagocytosis. When an antibody binds to its antigen, the stem region kind of changes conformation, and then it reveals a strong binding site for a receptor on macrophages, and the macrophages will gobble it up. Gone, gone, gone. They circulate for a certain period of time. If we're all done with them, they kind of fizzle out, and we'll find out later there's a way that the immune system can still be aware of what has come before, even though the antibodies don't last that long. An IgM antibody is a pentamer. It's just 5 antibodies connected together covalently. It's what's first released by plasma cells when you get an immune response going. And so they go into the blood and they circulate, and they're good at agglutinating. Look at all the valence, this thing. A whole bunch of different antigens can stick to it, whether they be cells or bacteria or whatever. And so they can all form huge clumps of antigen and antibodies because there's different binding sites. So they last only for a short time, and then the plasma cells switch over to making IgG typically. They start with IgM, then they class switch. It's called IgG. IgA antibodies are the ones that are secreted in mucus and whatever body secretions and in lacrimal fluid and so forth. So those are the guys that are present in there. They're dimers, 2 antibodies connected together by an attaching peptide. There's 4 different sites, binding sites, or a valence of 4. IgD antibodies, those are the B cell receptors. The B cells have antibodies all over their surface held down by the stem with the fork sticking out to just looking for antigens. And so they're basically antibodies, but they're called IgDs. And then if the B cell gets activated and goes on to produce plasma cells, it'll produce IgMs and IgGs with the exact same tips that were on that IgD. Remember, every B cell is unique, has a different antibody receptor with different tips on there. Immunoglobulin type E's work exactly the same way as immunoglobulin type G's, except that after the infection is resolved, the antibodies have been produced, they did their thing, clear the body of antigens. Immunoglobulin E's then adhere to mast cells and basophils and stay there. So now all your mast cells have immunoglobulin E's sticking out the surface, and the next time an antigen comes along into your body, that same antigen binds to that immunoglobulin E, mast cells release histamine and you have inflammation. So this is the basis of allergic reactions. Don't really want this kind of thing going on all that much. Okay, so how about some discussion of activation of B cells? So antigens enter the lymphoid tissue where the B cells are living, either in lymph fluid or when you saliva, when you eat something, it goes to your tonsils, or in your blood if it's in the spleen. So there's some lymphocytes, some B lymphocytes hanging around in large quantities in those lymphoid tissues, and the antigen will just sort of bounce off B cells until it happens to encounter one that recognizes it. Here's the magic thing about the immune system. The way the B cells and T cells are produced during development, by shuffling those sequences, the variable region of antibodies and the variable region of T cell receptors, you produce every B cell and every T cell that could possibly exist that could recognize any chemical in nature virtually. You produce an infinite almost number of different B cells and T cells. And then the so-called maturation process, you check every single one of those to see if they recognize a self-antigen, say a protein that's part of your own body's tissues and cells, and if it recognizes something like that, kill it. And what you wind up with is the entire range, mass number of unique B cells and T cells that could recognize any foreign antigen that could come into your body of almost any type whatsoever. That's what you wind up with. So you have just this unbelievable number of unique, different B cells and T cells. So the antigen is going to bounce off B cells for quite a while until it stumbles upon one whose receptor, whose IgD, recognizes that particular antigen that's infecting our body of that day, and that will activate that B cell. It starts to get activated. There are a couple of different types of antigens. There's a so-called, rarely, T-independent antigens. Some polysaccharides, once this happens, they bump into find that right B cell. The B cell becomes fully activated, proliferates into a clone, produces memory cells and antibodies, and there you have it. Done deal. Most antigens don't work quite that way. Most antigens, when they find their B cell, it's called clonal selection, incidentally. When they find their B cell, the B cell will become activated, but it's like starting your car, an analogy often used, and leaving it running in your driveway with the gear selector in park, and it's just sitting there running. But it's not ready to drive anywhere yet until a second signal comes along from a helper T cell, as we mentioned at the very beginning. Then you put that thing in drive and let it proliferate, form a clone, and some memory cells and stuff. So that's that second signal that's required. How does that all work? Well, let's just mention what are major histocompatibility complexes. They're complexes of glycoproteins that are on the surface of all the cells in your body. They must have it or they're doomed. Every cell in your body has to have MHC class 1 proteins on its surface. That's like having an ID card that says you belong in the body, you're cool with the body, and the body will let you alone. If you don't have it, if you're a tumor cell, for example, that doesn't play by the rules and doesn't make MHC class 1 anymore, it can be killed by natural killer cells, for example. MHC class 2 is a slightly different version of the same thing. Only so-called antigen-presenting cells of the immune system have MHC class 2. Macrophages, dendritic cells, and B lymphocytes. They are called antigen-presenting cells. When they take in an antigen, either by phagocytosis or being infected, they chop it up, they process it and stick it on the surface on MHC1 and MHC2, and that's what T lymphocytes are looking for. So helper T cells, when we have an actual infection in the body, not only will an antigen find its way to a B cell that's the right B cell, some antigen will find its way to an antigen-presenting cell, and that will activate a helper T cell, because the helper T cell is looking for a cell with MHC class 2. It's looking for an antigen-presenting cell. Once that antigen-presenting cell finds the right T cell, out of all the thousands and thousands of T cells, then that T cell will become activated and release cytokines that can turn on that B cell. Now it has full permission to go ahead and make plasma cells and antibodies. So here's a T cell independent antigen binding to a B cell, turns that thing on, it's going to go ahead and form a clone. Most antigens don't quite work that way. We need a helper T cell to be involved. So here's a diagram. Here comes an antigen into the body, some fragments. Perhaps there's been a war zone. There's some neutrophils doing battle with bacteria that are eventually getting filled with dead bacteria and dying themselves. And there's antigen fragments all over the place. Anyway, they come along and they bounce off B cells, clonal selection, until they find the right B cell, gets activated. It's like in the mood to go. It brings in that antigen into the cell and then displays it on its MHC class II. Because remember, B cells are one of the 3 antigen presenting cells. It has MHC class II. Where's my list? MHC class II is expressed on B cells, macrophages, and dendritic cells. So here's our B cell that has found its antigen, has its antigen proudly displayed right on the surface on MHC class II, and that's what helper T cells look for. They have a receptor that recognizes MHC class II. They just showed one thing. There's really 2 things there. There's a receptor called CD4 for that MHC class II, and then there's a T cell receptor that's unique on the tip, just like an antibody, that's going to recognize that antigen there. And if that happens, if we find that helper T cell that recognizes that antigen, that T cell will become activated, release interleukin 2. Interleukin 2 will turn on that B cell, then it will multiply into a clone, produce plasma cells and antibodies. And I must mention memory cells. One whole part of that clone, like if the thing produces, who knows, 10 million cells by cell division, a fraction of those are going to just go off and stay in the body as memory cells that will remain forever sort of pre-activated in case of a re-exposure, and then the other cells will go on to produce plasma cells and antibodies. This diagram finally says something about that. So here we have that whole idea I was talking to you about. There are just thousands and thousands of different unique B cells, each one with a different B cell receptor. See, the shapes are different. They just drew 4 to illustrate the point. Every B cell has a unique antibody receptor on its surface, and that antigen just bounced around until it found, in this case, number 3 happened to be the right one. Now that one is activated, and it's ready to go and proliferate, but it can't really proliferate without a second signal from a helper T cell. Once that comes along, it will start proliferating into a clone. Some of the clone cells will stay in the body as sort of pre-activated B cells. Those are called memory B cells. And the next time the same antigen comes along and comes in contact with one of those memory B cells, they'll be off like a flash, producing a response. But typically, the majority of the cells are going to differentiate into effector cells, they're called, cells that can do something about this problem. Plasma cells can secrete antibodies. So that's the humoral immune response right there in a nutshell. So as I was saying, during development, you produce just a myriad of different B cells, each with a unique receptor, some of which would recognize all your own body's proteins. Kill all those off, you're left with just the ones that recognize foreign antigens, and those are the ones that can be retained in your lymphoid tissues. How do antibodies help us anyway? Antibodies help us by these things. I'm going to show you in a picture because it's much easier than just talking about it. Here are the ways that antibodies can actually solve the problem of an antigen that's in the body that we don't really want around. Antibodies bind to bacteria, and because antibodies have multiple binding sites, they can form enormous clumps of antigen plus antibody, which can't really go anywhere. They're kind of big and they're clunky and they kind of fall into a pile, and then macrophages gobble them up and destroy them. It kind of traps all the antigens, all the bacteria in one spot. That's called agglutination. We use that in the laboratory, as we'll see later on, to do a bunch of cool tests for different microorganisms. All right, opsonization, we already defined that. When antibodies stick to the surface of an antigen, in this case a bacterium, the stem part of the antibody is changed so it binds to a receptor on macrophages. They gobble it up the most ferocious way. They might have a TLR that can recognize that bacterium, maybe not, but in this case, once the antibody's bound, then those macrophages will go after it like crazy, like having icing on a cupcake. And let's face it, without icing, cupcakes are not ready to eat. All right, how about another way? Activation of complement. When antibodies bind antigens, that is the classical pathway of complement activation. Complement gets turned on, we have big activation of inflammation, and we have opsonization happening not just with antibodies, but with complement proteins, but over here we have cell lysis, and that's going to kill any of these bacteria that are still surviving. They may have antibody on the surface, but now they have their membranes popped. And then antibody-dependent cell-mediated cytotoxicity, ADCC. Big old parasitic worm larvae are too big to phagocytose, but if antibodies are stuck all over the surface of them, along will come eosinophils and other and macrophages and so forth, and recognize that antibody on the surface and start secreting terrible, nasty, toxic substances that will hopefully kill that liver fluke or whatever it might be. So, finally, neutralization. Let's say you have a virus that can infect cells in your body, hide itself away, right? As soon as it gets onto the host cell, it dives down inside, and now it's hidden from view. That would give it time to multiply. Well, antibodies, once they're produced, anytime some virus particles are released, antibodies stick to them. Now they can't bind to their host cell receptors. They can no longer be internalized. They can no longer infect any cells. Excuse me. That's called neutralization. Or what if it's a toxin? We said that some exotoxins are extraordinarily poisonous, very, very potent. Only a few toxin molecules can kill. But if there's antibodies present, such as something you've been vaccinated against, such like diphtheria toxin, or what's another one? I'm trying to think of another example that you get immunized against. Toxoid vaccine. Anyway, we have toxoid vaccines, which denatured toxins have been used to produce antibodies, and we immunize people with those so that they have antibodies. And if that toxin does come into your body, tetanus toxin won't cause paralysis. Why? It'll be neutralized immediately by antibodies. All right. How about T cells and cellular immunity? That's all about humoral immunity and a little bit about T cells, because the helper T cells were required to get the B cells fully geared up to do their thing. So in this case, we have cells that are a little more complex because they have to have two receptors working. One has to recognize a MHC class 1 or 2. There's two populations of T cells that are separated by those two different things. So they have an MHC receptor, and they also have a separate T cell receptor, which is that unique receptor that every cell has that's different, that can find a particular antigen of the day. So we take those cells from the red bone marrow, send them to the thymus to make sure that, number one, they have the MHC recognizing receptor, because if they don't have that, they're screwed up. Get rid of them. Once we know they have the MHC receptor that's correct and checks out okay, then we check and see if they recognize any self proteins. If they do, kill it. And we wind up with, just like with the B cells, we wind up with all the possible T cells, each with unique receptors that would recognize MHCs, but also unique antigens because of their T cell receptor. Only foreign antigens, though. We've got rid of all the cells that would recognize self. So that's a very important process in the thymus gland. Without that, any mistakes in either one of those maturation processes, we have the potential for an autoimmune disease, because we might have a cell that just a shade of recognition of a self protein can lead to a full-blown immune attack on our own body. All right, so we have helper T cells. We've already talked about those a little bit. We have cytotoxic T cells, which we're going to get into a little bit more. And then we have regulatory T cells and so forth. So it turns out that helper T cells produce different cytokines depending on what cytokines were around when they were first activated. And so-called Th1 T cells produce, there's another slide coming up about this, but produce gamma interferons as well as interleukins, and those tend to activate cellular immunity, cell-mediated immunity. They activate cytotoxic T cells and natural killer cells and macrophages, whereas other helper T cells produce cytokines that more are geared towards activating B lymphocytes, and they produce immunoglobulin M and G and occasionally E. Cytotoxic T lymphocytes are the ones that are going to go around later and kill infected cells in your body once they're activated. All right. So T cell receptors are what are the unique receptors on T cells, and they have to be clonally selected. We have to find out where is that T cell who can help us out on this particular day the antigen that we have. Antigen-presenting cells, again, are macrophages, dendritic cells, and B cells that can take in antigens, process them, and then stick them on the surface on their MHC complexes. Interestingly, just a little aside here, pathogens in the lining of the intestines or the respiratory tract can pass through microfold cells. It's like a little portal of entry into some lymphoid tissue. If it's in the small intestine, the later part of the small intestine is called a Peyer's patch. And in that Peyer's patch are antigen-presenting cells, phagocytes, and B lymphocytes, and so forth. So we're going to get a sample. Here comes a pathogen, and it's going to get endocytosed by this M cell, and the M cell is going to transport it right across, where it's going to be thrown into a pit of alligators. It's going to bump into some lymphocytes who will be clonally selected and activated. An antigen-presenting cell will probably gobble it up and express some antigens on its surface, and that will activate some B cells. Get my cursor going. We'll activate some helper T cells to let the antibodies be produced. There might be some cytotoxic T cells around that will get activated. So this is a way that we can transport the antigen, the pathogen, right into a nest of immune tissue and all the cells that are capable of doing humoral and cellular immunity. All right, so more specifically, helper T cells are a population of T cells that express a MHC recognition receptor called CD4. CD4 recognizes MHC class II, so helper T cells are only concerned with dendritic cells, macrophages, and B lymphocytes. That's all they check. They go around checking for MHC class II. If they see MHC class II, then they say, let me see if my T cell receptor recognizes the antigen that's being displayed on there. If so, it becomes activated, and it turns out that antigen-presenting cell can express a new receptor on its surface, which then can bind to a receptor on the helper T cell and complete the activation of the helper T cell. Also, the antigen-presenting cell could release interleukin-1, which will complete the activation of the helper T cell. Now the helper T cell is ready to go. It starts releasing cytokines that can turn on B lymphocytes and cytotoxic T cells. But again, in every case, the antigen-presenting cell might not be producing the same cytokines, and will have different helper T cells produced, some of which will produce mainly interferon gamma and interleukin-2. Oops, an M got there. A little typo I didn't notice. Oops. I'm not the most reliable typist. Wait, I don't know which. Anyway, and those guys activate cellular immunity and innate immunity. Th2 helper T cells tend to produce cytokines, which will activate or act as growth factors for B cells in producing immunoglobulins, antibodies, so humoral immunity. There's a bunch of different helper T cell types we won't go into. They find out new stuff about the immune system about every week. But anyway, Th17 cells are cells that are more capable of stimulating aspects of innate immunity. I'm not sure what TF are in this particular scenario. I'll just have to look that up. It's an abbreviation I'm not familiar with. All right, so anyway, a little review. Here's our B cell. We have clonal selection. We have activation. We start producing some antibodies. And a helper T cell bumps into that B cell with its antigen, becomes activated, and it's going to produce either a Th1 response, gamma interferon, and interleukin-2 perhaps, an interleukin-2 or 4 that's going to activate B lymphocytes and possibly activate some mast cells and neutrophils and macrophages. All these cells are going to get activated by a helper T cell by cytokines that it produces. So you can see the cytokines are super important. There's so many different cytokines that do different things in different scenarios, and we're not going to get too hung up on that. Just so you know that there's quite specific and have specific tasks and roles that produce the complex, nuanced immune response that we see in our bodies. So here's a little diagram showing an Antigen-presenting cell takes in some antigen by phagocytosis in this instance, processes it, meaning it fuses it with a lysosome, breaks down some of the glycoproteins and proteins and so forth, and takes fragments and places it on MHC class 2 and then displays it on its surface. Antigen display, it's called on it. Antigen, that's antigen presentation. And here's a helper T cell whose job it is to go around looking for cells that have MHC2 on their surface using its CD4 receptor. I don't know why they didn't bother to draw two receptors here, right? We just got done explaining that they do have CD4 on their surface. So CD4 recognizes the MHC2. The T cell receptor checks, is that antigen a match? If it is, yes, that cell gets activated. The antigen presenting cell can produce a co-stimulatory molecule or receptor that could then bind to the T cell and activate it, or secrete a cytokine or interleukin that could activate it. Point is, the helper T cell is now completely ready, co-stimulated, ready to go, and it can now release cytokines that can activate humoral immunity and cellular immunity. CD8 lymphocytes are activated by MHC1 with antigen on its surface, and antigen-presenting cells can display antigens not only on MHC class 2 but on MHC class 1, but also any cell in your body that gets infected can display antigen fragments on MHC1. All of the cells in your body, including antigen-presenting cells, have to have MHC class 1, and that's what the CD8 receptor is all about. CD8 plus T cells are those that have the capability of recognizing MHC class 1. So that's what they're looking for. Antibody cell. They look for the MHC class 1, check it out, looks good, everything's right, looks like it's properly expressed. Then they check with their T cell receptors, is there an antigen there, and if so, do I recognize it? If so, it becomes activated. Once it's fully activated and has received a second signal from a helper T cell, then it can kill that infected cell. Cytotoxic T lymphocytes, CTLs, that's what they're talking about here. They proliferate when they're activated, producing memory cells, right? B cells produce memory cells, helper T cells produce memory cells, and cytotoxic T cells. So anyway, they produce memory cells, but also effector cells. Effector cells are the ones that could go around. From then on, they just surveil the body looking for infected cells. Perforin is a protein that they secrete that can puncture the membrane of an infected cell of your own body, just like the membrane attack complex. And then granzymes are poison that goes inside the infected cell and triggers apoptosis, and the cell dies. So these are bad blank cells. They can go around killing cells of your own body. That's a non-trivial issue. They're quite powerful and almost dangerous in a way. But that's what they do. They prevent hidden antigens hiding in your infected cells from being able to continue to spread. So there's that MHC class 1 with an antigen expressed on its surface, and here's a T cell with its T cell receptor. It's in this case a CD8 positive T cell, and it recognizes therefore MHC class 1. Checks antigen. Yep, it matches my antigen receptor, becomes activated, and now it can go around the body looking for infected cells that also express the same antigen on their MHC class 1. They should have drawn that in there. The cell will have MHC class 1, and that's where it'll express that antigen, and the cytotoxic T cell will then kill it. Another artistic rendering, very delightful. There are some T cells called regulatory T cells, which suppress T cell activity. It's thought that maybe those cells are involved in preventing accidental attack on self cells of the body, or they may be what helps sort of settle down the adaptive immune response after a certain number of days. We don't want this going on indefinitely. This is a storm of cytokines and bad cells going around just killing, crushing, and destroying everything. And so we got to keep things under control. Antigen presenting cells, what do? They digest antigens, display them on their surface on MHC1 and MHC class 2, and they include these three cells, which we've mentioned several times already. MHC class 2 with antigen displayed is what turns on helper T cells. MHC class 1 with antigen displayed on it is what activates CD8 plus cytotoxic T cells, and that's humoral immunity is antibodies and B cells. Now we're talking about cellular immunity with those cytotoxic T cells. Natural killer cells are basically like a cytotoxic T cell, but they don't go through this clonal selection activation process. They get activated by gamma interferon and other cytokines when there's a big inflammatory response going on, and they go around looking for any cells that don't express their MHCs correctly or don't express them at all or have some other thing about them that the natural killer cell recognizes as foreign, kill it. And they kill cancer cells, for example. They can attack parasites, but the thing is your immune cell system kills off tumor cells which are produced during your body throughout your lifetime. And you don't know about it. You never know about it because it's always a race between tumor cells trying to grow unabated and beating out the immune system and being able to form a tumor and getting some blood vessels to grow in and so forth, versus the immune system trying to shut it down, kill it off, and most of the time the immune system wins and kills that tumor. Sometimes, though, a cancer can develop. People that get immunocompromised have all kinds of tumors because they no longer have that immune system that's there always checking for any abnormal looking cells and killing them off. Again, antibody-dependent cell-mediated cytotoxicity we mentioned before. Here's a larval cell, larval multicellular worm thing from a parasite helminth of some kind that's in the body, and it's getting antibodies all over it because some antigens have been discovered, have been activated the humoral immune response, and now these eosinophils and macrophages recognize that FC region on the stem of this activated antibody and start releasing terrible, nasty chemicals and try to kill that thing off. And there's a liver fluke with just coated with eosinophils that are releasing toxic substances to kill that fluke. Cytokines, you can have too many cytokines. If you have too many cytokines, for example, when you have a super antigen, then you have a problem because there's too much inflammation. It can cause massive vasodilation. One of the things that we said about inflammation is histamine is released and it causes vasodilation. Well, if you haven't studied physiology yet, I hope you will soon. If you have too much vasodilation in your body, your blood pressure will tank and you won't have enough blood circulating to meet the needs of all your tissues. And you can actually die from a cytokine storm from excessive vasodilation and a drop in blood pressure. So just a couple of cytokines. Interleukin-1 is a major inflammatory cytokine released by activated macrophages. They also release tumor necrosis factor alpha. Interleukin-2 is released by helper T cells to activate the proliferation of B cells and cytotoxic T cells. IL-12 activate Th1 responses, gamma interferon, and so forth. TNF-alpha, again, released by activated macrophages, promotes inflammation. Hematopoietic cytokines, some cytokines that are released during all this activity going on causes the bone marrow to produce a lot more white blood cells. So all the while these things are taking place, a whole bunch of new neutrophils are coming into the circulation to help fight against whatever infection we have so that they can leave the circulation, diapedesis and so forth, and travel by chemotaxis to the site of infection and kill. And so that's because of hematopoietic cytokines that go to the red bone marrow and turn things up a notch. Interferon alpha and beta, we said, are what help neighboring cells to an virally infected cell shut down so they can't produce any new virus particles. Gamma interferon activates macrophages and natural killer cells and so forth. So, all right, one more really important topic I think we have left to cover, and that is primary versus secondary immune responses. I know we're going a little bit long, but there's got to finish up some really important things to understand about vaccines. So here is a graph showing the concentration, or titer it's called, of antibodies in the blood versus days of time after exposure to an antigen. So over here at day 0, we're going to get exposed to an antigen, right? And it's going to start clonally selecting, searching for the right B cell. Then a helper T cell has to get activated, and time is going by, days are going by. Finally, you got to start producing a clone of B cells, which takes a while. Then they got to start maturing into plasma cells. All this takes about a week. Look at that. A week of time goes by before a lot of immunoglobulin type M winds up in the circulation that can actually do those 5 things we saw to help remediate this problem of this antigen: inactivation and opsonization and agglutination and precipitation and all those things that antibodies do. And then finally, there's class switching. The plasma cells switch over to making immunoglobulin G. So look at how long it takes, maybe 2 weeks to reach the peak antibody titer in your blood after an immune exposure or an antigen exposure. Okay, so now we let everything settle back down. We said the antibodies don't last that long in your blood. They begin to go, the titer goes back down again. Maybe years go by, who knows, however long it might be. And then get re-exposed to the antigen once again. Second exposure, or however many times again, we get re-exposed to the antigen. What happens? The memory cells that were left behind in the body bind to that antigen and immediately start proliferating and differentiating and producing effector cells rapidly. So here's our second exposure right here. And if we can somehow keep track of the scale of this diagram, about a day or 2 days later, we're starting to already see a titer, a significant titer, in fact, a larger titer of antibodies coming into the blood than before. Significantly more antibody, much more rapidly than the first exposure. This is the power of memory, immunological memory. It's not some ethereal thing like when you remember something that happened in the past. It's physical cells, B cells, T helper cells, and cytotoxic T cells that have remained behind from a previous exposure to an antigen, and they are ready to rock. And that's why if you get a vaccine, you produce this primary immune response, this first immune response, and memory cells remain behind in your body. And if you're exposed to the antigen once again, the actual pathogen in this case, perhaps, you won't get sick because this reaction is so fast and so vigorous, the antigens will never, the bacteria will never get a chance to grow to a sufficient quantity to produce disease. So when you get exposed to an antigen, all these things happen, all those things we've been talking about all chapter long happen. That's called active immunity. You produce the antibodies, you produce the cytotoxic T cells, and you produce the memory cells. Naturally acquired active immunity is what happens when you get sick or you get exposed to a cold virus or whatever it might be. Passive immunity, on the other hand, is when you acquire antibodies by themselves. Instead of an antigen coming in your body, you just receive some antibodies from somewhere. That happens on a natural basis across the placenta. The fetus receives antibodies from the maternal circulation. That's called passive immunity. Those antibodies still work. They can still, if that newborn gets exposed to an antigen, starts to cause an infection in his body, those antibodies can do all the good things the antibodies do. So they're quite robust when they're born. However, then those antibodies kind of disappear from the circulation relatively rapidly. And then babies become very, very susceptible to disease because they don't have any memory cells. Passive immunity does not produce memory cells because all we received was the antibodies from somewhere else. Okay, how about artificially acquired active immunity? That's what vaccines are. We intentionally produce a vaccine that will elicit an adaptive immune response against some antigen that we know could produce a very terrible disease, and then later on, if we actually get exposed to that pathogen, diphtheria, bacteria, or something like that, we won't actually get diphtheria. Why? Because we have memory cells that can go to town and whip those bacteria before they ever have a chance to colonize the bodies appreciably. So yes, we have memory cells when we use vaccines. Artificially acquired passive immunity, we said naturally was the placenta or breast milk. That's one of the reasons that people advocate breastfeeding, is because immunoglobulin Gs are in the breast milk and can go right into the baby's gut and go into the bloodstream and continue to produce immunity for a period of time, much longer than if they just had the antibodies they were born with. Anyway, artificially acquired passive immunity. Let's say someone gets a snake bite and they're going to die in a few hours. What do? Wait for them to produce an adaptive immune response? Probably not. The 5 to 7 days time it takes for that to happen would obviously be not even close to effective. We're going to inject them with some antitoxin. Remember at the very beginning we talked about the Nobel Prize for antitoxin. We keep in the emergency supply kit, we have some snake venom antitoxin, meaning an antiserum produced in animals, and we've purified the antibodies, and now we can inject that right into someone's body, sweep away that venom, their macrophages in their liver, for example, will gobble up the antibody antigen complexes and they'll be saved. Thing is, there's no memory cells, so if they get bit by the snake the next day, sorry, right back to the same emergency room for another injection of antitoxin. All right, serology is the study of reactions between antibodies and antigens. We'll talk about applications of serology for diagnosing disease. Where'd my slide go? There it is. Antiserum is just plasma that has been allowed to clot, centrifuge out the blood clot, and all the antibodies are still floating in there. And now we have an antiserum, which is essentially an antitoxin, or whatever you want to call it. And we can use that antiserum for any number of things, including lots of diagnostic tests to diagnose disease. Immunoglobulins or gamma globulins are just the names for antibodies. And that's just a little cartoon of an electrophoresis system to when they first started separating plasma proteins from each other by electrophoresis, by putting them in an electric field in a type of a gel matrix, just like kind of like the agar we use for growing bacteria in the laboratory. They saw that they all had unique properties and stuff. We don't need to really worry too much about that. Anyway, thank you for bearing with me for kind of a long chapter, but a wonderful chapter, almost magical in the way the immune system works. And then we'll spend a little bit of time talking about ways that microbiologists have co-opted some of the tools from the immune system, such as antisera and so forth, to create tests for different pathogens that are in people's bodies at a given